The overall goal of this paper is to demonstrate the potential of PTR-ToF-MS for direct volatile organic compound analysis in food science and technology, and in particular, the importance of automated sampling and tailored data analysis.
Volatile organic compounds play a key role in food science and technology, both because they control flavor and odor perception, and because they provide a tool for the monitoring of food quality. The high sensitivity and the rapid and direct injection make PTR-MS one of the best choices for the monitoring of volatile organic compound release, for bioprocess monitoring, for the screening of large data sets, and for nosespace analysis.
We also develop proper data handling and analysis tools which are necessary for proper exploitation of large and complex datasets, such as the ones produced by PTR-MS in a very short time.
Visual demonstration of our methodology can inspire researchers from different transdisciplinary scientific fields, such as sensomics, metabolomics, breath analysis, and phenomics.
To begin the first experiment, add 5 milliliters of pasteurized milk to 20 milliliter glass vials equipped with PTFE silicone septa. Then, transfer the samples to a multifunctional GC autosampler equipped with a temperature control tray. Use the robotic arm of the autosampler to inoculate the vials with the microbial starter cultures. Set the incubation time according to the typology of the desired process and the specifications reported by the starter culture manufacturer. Then, set the autosampler to analyze one sample after the other, obtaining an online volatile organic compound monitoring